Okay, so here we are still in chapter 17 on lipids. It's one of our longer chapters, so hopefully it's been keeping you interested and doesn't feel like it's dragging on for you. Certainly never feels like that for me because it's also interesting, the chemistry and the biological chemistry of these lipid molecules. So we've talked about a number of different lipid types so far, and we've looked at the more traditional triglycerol-based lipid types and even the sphingosines, the sphingolipids based on sphingosine. So now we're ready for a very different type of lipid. All right, you've been waiting for it, right? Now we're going to discuss the steroids, which don't have well, they don't necessarily have that ester linkage that we've seen in most every other lipid molecule we've discussed so far. So instead, we're going to look for that fused ring system, the four fused rings together, which is our basis of cholesterol, and then the steroid hormones that are similar in structure to that cholesterol parent. Okay, so what are you looking for to distinguish a steroid from the other lipid types we've talked about? Well, mainly you're looking for that fused set of rings. So steroid molecules contain a steroid nucleus with 3 fused cyclohexane rings, and then at the end we have 1 cyclopentane ring. There are no fatty acids, and there's a total of those 4 rings, A, B, C, D, as we have them lettered down below. The steroid nucleus is numbered from the carbon atoms in ring A, which is our furthest left cyclohexane ring, and then 2 methyl groups at 18 and 19 there, as you see our numbering system. So that's your basic steroid nucleus and the steroid numbering system that we'll build on for cholesterol and then some of the steroid hormones that we'll mention in this lecture. Okay, so cholesterol is our most important and most abundant steroid in the body. It's a sterol because it contains that oxygen atom as a hydroxyl group and OH on carbon 3. There's also a double bond between carbons 5 and 6, and there are methyl groups at carbon 10 and 13. And finally, it has a carbon chain at carbon 17, if you refer back to the previous slide for that numbering system, if you haven't already committed it to memory. I'd be pretty impressed if you had. So that's our basic cholesterol, and again, we hear a lot about it being bad, but that's mainly because in the American diet we tend to overconsume it, and many of us overproduce it as well, or don't use it correctly, however you want to think about that. So there we have cholesterol, an important and abundant steroid, and a necessary steroid, as we'll discuss. So here we see the structure, and it has those features that we just mentioned. So that fused steroid ring, the 4 rings, the 3 cyclohexane and 1 cyclopentane ring, and we see the double bond, the methyl groups, the carbon chain up there at the top of the molecule, and then the OH group at the bottom left to make it a sterol. It's a component of cell membranes, myelin sheets. We talked about myelin in the previous lecture and its importance. And then finally, brain and nerve tissues, again, an important component of those brain and nerve tissues, helping to insulate and improve the conduction of the electrical impulse in nerve cells. So cholesterol is good, it's necessary, it's useful. It can also be bad if there's too much of it. So cholesterol in the body is normally obtained from animal sources, meats, milk, and eggs, and it can also be synthesized in the liver. It will clog arteries when you have high levels that form these plaques. So we see on the right-hand side a normal open artery, and then we see down below a very unpleasant clogged artery because of excessive cholesterol plaque. So again, it's not that you can totally eliminate cholesterol either from the diet, and even if you did so, it would be synthesized in the liver, but you should hopefully be able to have a healthy, balanced diet that includes enough cholesterol without excessive amounts, as well as, again, what your own liver does and what your family history and how your particular genetic predisposition to dealing well with cholesterol or not dealing well with cholesterol hopefully will keep you looking and finding healthy balance between cholesterol intake and then what your body does with that cholesterol. And so here we see one of the problems, and that's the cholesterol in foods tends to be about 400 to 500 milligrams of cholesterol per day for the typical American. The American Heart Association recommends no more than 300 milligrams of cholesterol per day. So that's, again, less than most of us are consuming. That's a problem. Saturated fats in the diet may also stimulate the production of cholesterol by the liver. So even if you are getting less than the 300 milligrams that's recommended for cholesterol itself, if you're taking in a lot of saturated fats, you may be producing more cholesterol in your liver than is healthy or good for your body. And we can see if you eat a 3-ounce serving of liver, beef liver, you've already taken in more cholesterol than the American Heart Association would like you to take in a single day. Doesn't mean you shouldn't eat liver. Liver's got lots of good redeeming qualities, but it means you should be careful not to overindulge or eat it too frequently. Then we see eggs again. One large egg has 200 milligrams. That's quite a bit of cholesterol, but again, there's good high quality protein there. It's all about a balance, in my opinion. And you see some other foods, lobster. Again, I think most of us probably couldn't afford to have a lobster a day, but if you can, you probably shouldn't anyway, because 3 ounces of lobster has quite a pack punch of cholesterol, 175 milligrams there. Fried chicken is another high cholesterol food. Then we go down in amount to essentially no cholesterol in margarine, although we talked about margarines as potentially having some unhealthy trans fats depending on how they're made and what they would disclose on their label. So again, all things in moderation, that's my opinion, but certainly not a dietitian or a healthcare provider. So certainly you should discuss your diet with an expert if you're concerned about the cholesterol either that you're taking in or that your body may be synthesizing. Let's go ahead and use this opportunity to do a learning check on the cholesterol molecule as well as your understanding of organic chemistry to date. So here you're going to match the components of the cholesterol molecule with each of the following types of groups or things. So pause the video here while you make those determinations. So use the letters A, B, C, and D from the diagram below to determine which is the alkyl chain, steroid nucleus, hydroxyl group, and methyl group. Once you've made those determinations, start the video back up and we'll see how you did. Good luck. Okay, so hopefully this was pretty easy for you. I would imagine at this point in the semester, at least the alkyl chain should be pretty easy to pick out there. It's part D on our diagram. The hydroxyl group is at the other end is part A. And then the steroid nucleus, that fused set of 4 rings, is C. And finally, the methyl group there was denoted by B. That might have been a little tough to pick out what B was actually looking at. It was just the methyl group, not any of that ring there. So hopefully you were able to make those determinations pretty easily. If you did make any mistakes, hopefully now it's clear which regions were identifying which components. And if that's not the case, then certainly please reach out to me and let me know if there's anything I can do to help clarify things. So in addition to cholesterol's role in the body, there's also these bile salts that are synthesized from cholesterol in the liver. Then they're stored in the gallbladder and finally secreted into the small intestine. They have polar and nonpolar regions, just like a lot of these lipid molecules we've discussed, and they help to absorb cholesterol into the intestinal mucosa. So sort of something synthesized from cholesterol with the aim of helping absorb more cholesterol, an interesting sort of body mechanism there. But again, the bile salts are necessary and helpful for that reason. If you don't have a gallbladder, then they would travel directly from the liver into that small intestine for additional absorption of cholesterol. Here we see an example of a bile salt. This one happens to be sodium glycocholate. And you can see there it has a nonpolar region, the cholesterol nucleus there, the steroid nucleus, I should say, and it has that OH group there at the bottom left, just like cholesterol would. But we also see that we have a polar region here from glycine, so that's an amino acid that we'll talk about in a couple chapters from now, and that gives it a nice polar region. The nonpolar region, of course, is similar to cholesterol, but it's actually from cholic acid, which is a bile acid, and that's how we end up with this sodium glycocholate. And again, the sodium salt is because we've taken that amino acid and we've neutralized it with, say, sodium hydroxide for simplicity's sake. Now, one of the problems involving those cholesterol-based bile salts can be gallstones, and gallstones are formed when large amounts of cholesterol accumulate in the gallbladder. They're composed of almost pure cholesterol, 100% cholesterol nearly, with some calcium salts, some fatty acids, and some glycerophospholipids. And we see here some gallstones that would cause quite a problem, and unfortunately it does afflict a fair number of people, and then the gallbladder is often removed as a result. And like I said, the gallbladder it's good. It's good to have if you can. It helps to concentrate those bile salts, but if you need to have it removed, the bile salts can flow directly from the liver into the small intestine, because as the gallbladder is removed, that pathway is surgically allowed to be maintained. Now, in the early days, it was just the cholesterol levels that we were concerned with, and as I said, those can be based on diet or from heredity, depending on how your particular liver based on your genetic fingerprint deals with those fatty acids and making cholesterol. So fortunately, we now understand that it's not just the cholesterol levels itself, it's the lipoproteins that are also important. And these are spherical particles. They have an outer surface of polar proteins and then some glycerophospholipids that surround hundreds of nonpolar molecules of triacylglycerols and cholesterol esters in the inner portion. They combine lipids with proteins and phospholipids. So when we talk about proteins in a couple chapters, that'll make a little more sense. They're soluble in water because the surface contains those polar lipids. Polar proteins, I should say. So we have the different regions. There's the polar proteins and there's the polar ends of the lipids themselves that allow that water solubility. This is how we can do blood tests, because if it's water soluble, then it's going to be blood soluble, and the lipoproteins are quite easily isolated from the blood. So here we see an image of a lipoprotein, and so inside that lipoprotein or lipoprotein, however you prefer to pronounce it, we see there's lots of those nonpolar lipids. So inside we have things like the cholesterol esters, the triacylglycerols, the things that are really largely nonpolar and would not do well out in the bloodstream by themselves. They're packaged nicely in these spherical lipoproteins. The surface of the lipoprotein to allow that blood solubility. We see there's membrane proteins that are highly polar. There's the glycerophospholipids, at least the polar ends of those, the head groups. And then cholesterol, we see that OH group as a polar region of the cholesterol that allows for it to interact well in the bloodstream with all that water, whereas the nonpolar tail of those lipids, or the nonpolar ring system of the cholesterol, are pointed in toward those nonpolar lipids, the triacylglycerols and the cholesterol esters. So that's how the body does it. It's pretty ingenious. Biological chemistry is a really neat application of chemistry, because you've got to deal with the fact that we have water everywhere, but we need to have regions that are nonpolar as well, and lipoproteins is one way that biology has solved that problem, by containing all those nonpolar entities inside this spherical lipoprotein with a surface that is quite polar. So there are different types of lipoproteins, and they differ in density, composition, and function. And so they include the chylomicrons, the very low density lipoproteins, VLDLs. There's the regular low-density lipoproteins, LDLs, which carry cholesterol to the tissues. And then there's the high-density lipoproteins, HDLs, that pick up cholesterol from the tissues and carry it to the livers. So we'll talk about these different types in detail, but probably already it makes sense that the HDLs are good because they're picking up the cholesterol from the tissues and carrying it to the liver, whereas the LDLs are carrying it from the liver to the tissues, and so that's probably associated with things that might lead to that cholesterol plaque buildup in the arteries. So the LDLs are important. They are necessary. They transport that cholesterol to tissues where the cholesterol can be used for synthesis of cell membranes and steroid hormones. They deposit cholesterol in the arteries in the form of plaque when those cholesterol levels exceed the levels needed by the tissues. So it's not that LDLs are bad, although they are called the bad cholesterol because of that contribution to heart disease. They're only bad when they're in levels that are so high that you get that plaque formation, right? The plaque formation is bad, not bringing those cholesterol molecules to tissues where it can be used for synthesis and for steroid hormones. So just be careful. LDLs, you don't want to have 0 for your LDLs, but you also don't want to have too high a number because the high LDL count is what's associated with cholesterol level that forms plaque in those arteries. Now the HDLs, they're high density lipoproteins, and they're more dense than the LDLs because they have more protein present. So lipids are low density. They don't pack well, as we've talked about for a number of lectures now, and therefore they tend to be lower density. Here we've got a lot of protein as well that does pack nice and tight, and we get a higher density lipoprotein here. As we mentioned earlier, they pick up cholesterol from the tissues and carry it to the liver where it can be made into these bile salts that ultimately get eliminated from the body. And as a result, they're called good cholesterol because they're seen as taking that excess cholesterol and getting rid of it ultimately by allowing it to be ejected, however you want to think about it, leaving the body with the feces. The VLDLs, the very low density lipoproteins, carry those triacylglycerols synthesized in the liver to the adipose tissues for storage. So these are very low density because they have a lot of triacylglycerols in that inner portion, the nonpolar portion of that sphere. This is a good thing, right? We do want to have some stored reserves of fats, but of course, in the American diet, we tend to overconsume, and so many of us tend to have extra adipose tissue, more fat cells than we really want or need, and so these can also be associated with artery plaque buildup. So again, the LDLs and the VLDLs tend to be your bad cholesterol types, at least for um, the lipoproteins, and then the HDLs tend to be your good, but of course we do need certain levels of all. It's just having too high levels of the LDLs and VLDLs that is associated with plaque formation. Those of you headed into the health professions will undoubtedly encounter lipid panels, and this is a type of blood test that measures serum lipid levels. It includes plain old cholesterol, the triglycerides, and then also levels of HDL and LDL, and the results of that lipid panel are used to evaluate a patient's risk of heart disease. Notice it's all 4 of those that are considered, and certainly as we learn more, we'll probably be interested in more particulars of this process, but again, it's not just the cholesterol, the serum cholesterol level anymore. It's really how all these pieces fit together and what's your relative HDL, LDL levels rather than just absolute values for any of these things. And so here are some typical recommended levels and then levels that would result in greater risk of heart disease. So total cholesterol, ideally it's going to be less than 200 milligrams per deciliter. Greater than 240 puts you in that higher risk category. The triglycerides, or the triacylglycerols as we've referred to them, they're less than 150 milligrams per deciliter is recommended, and once you get over 200, then you're at greater risk of heart disease. The HDLs, we want greater than 60 milligrams per deciliter. If it's less than 40, that puts you in a danger zone there. And then the LDLs, we want less than 100. If it's greater than 160, then you're in trouble. But again, it's really what the HDL level and LDL level tell is more important than just one or the other alone. And then finally, the cholesterol to HDL level, we want it less than 4. If it's greater than 7, then that's an indication that you might be at increased risk of heart disease. So a lot of it can be treated with diet. Some of it, there's a number of medications that can combat that issue if you just happen to produce a lot of cholesterol, or just based on heredity, you have low HDL levels, high LDL levels, but it tends to be a combination of heredity and diet that tend to land people in those higher risk categories for heart disease. So the other important use of steroids, in addition to the very important cholesterol and lipoprotein use, we've got the steroid hormones, and these are chemical messengers in cells. They're the steroid hormones, at least, are produced from cholesterol. That's the parent structure. They include the sex hormones, such as the androgens, the male sex hormone like testosterone, and then the estrogens, or estradiol, in females. So these are some pretty important steroid hormones, and they're based, as you'll see, on the structure of cholesterol itself. Okay, so we see here a few of these steroid hormones. There's testosterone there that's produced in the testes of males. It's also produced in females, so it's not solely produced in the male's testes, but you can see its structure there. We see we have a ketone functional group there, where in cholesterol and estradiol we have the OH group. Notice estradiol is an aromatic ring there for that six-membered ring that has the OH group, whereas it was a non-aromatic ring in the case of the other hormones here, and in case of cholesterol itself. So they all have that fused ring system, the 4 total rings, 3 six-membered rings and a 5-membered ring. We see progesterone up at the top right that's produced in the ovaries along with the estradiol. And then we have norethindrone at the bottom right. That's a synthetic progestin, and it's used in the oral contraceptives, the birth control pill, so to speak. Is typically, that's one type of synthetic progestin that might be present there. So there we have 4 examples of steroid hormones based on a cholesterol ring system, a cholesterol nucleus. In addition to those sex hormones, we also have the adrenal corticosteroids, and these are steroid hormones that are produced by the adrenal glands, those glands that sit on the top of each kidney, and they can include things like aldosterone, which regulates electrolytes and water balance by the kidneys. You've also got cortisol, which is released under stress to increase blood sugar and regulate carbohydrate, fat, and protein metabolism. And unfortunately, in our stressful life here in modern American society, we can also have increased cortisol levels at unfortunately a long-term rather than short stresses like humans were probably subject to, biologically speaking, in terms of evolutionary stresses when we were still hunters and gatherers and all that stuff. Nowadays we have things like work stress and family stress that can result in increased cortisol levels in the long term. And finally, we have prednisone, which is a synthetic corticosteroid. It's ultimately derived from cortisone, and it's often prescribed for reducing inflammation, treating asthma. If anyone's had asthma or have had family members with asthma, you probably have dealt with prednisone and its side effects. And then finally, rheumatoid arthritis is another place where this synthetic corticosteroid is used medically to help treat that condition. So here we see the structure of those adrenal corticosteroids that we just mentioned. So cortisone, cortisol, prednisone, and then aldosterone, which is a mineral corticosteroid. Or a mineralocorticoid, as it's called here on our slide. And you see they're all very similar in structure, although they can have fairly distinct and important chemical properties in the body. All right, let's get back to our usual pattern of ending with a learning check. So this learning check asks you to identify each of the following as a steroid, triacylglycerol, phospholipid, or sphingolipid. So for each of those things, A, B, C, D, E, F, G, go ahead and identify which class it belongs to. Obviously, you'll have to use a couple of these more than once. And once you've made your determinations, please start the video back up and we'll see how you did. Good luck. Okay, so cholesterol should be pretty fresh in your mind. Hopefully you correctly identified that as a steroid. For B, glycerol, 2 fatty acids, phosphate, and choline, that's a phospholipid. In particular, that's a lecithin because of the choline. We've got C, glyceryl tristearate, that's a triacylglycerol, or a triglyceride as it's commonly called in medical terminology. We've got D, sphingosine, fatty acid, phosphate, and choline. That's a sphingolipid. Hopefully the sphingosine gave that one away for you. Then we have estradiol, which if you remember, that was one of our steroid hormones. F, the bile salts. Those are also steroid based on cholesterol. And then G, lipids in plasma membranes tend to be the phospholipids and the sphingolipids. We mentioned previously that both of those are found in the plasma membrane. So hopefully you were able to get all those correct. If you did miss any, hopefully now you're able to see why the answers given here are the correct choices, and if that's not the case, and if you go ahead and try this some more on your own and continue to be frustrated, please do reach out to me for some help. Otherwise, we'll see you in the next and last lecture of Chapter 17.